basically, you would take two individuals that had the same phenotype, homozygous true breeding, recessive mutations, and you would cross them. And in the very first F1 generation, you would ask what is the phenotype of that individual. So, if you had two mutations that were generating white eyes, you would basically ask, if I cross these, what happens? So we're going to look in the F1 generation basically, and so you take a true breeding that's white-eyed with a true breeding white-eyed, and you ask in the F1, is this individual white-eyed or red-eyed? And if it is white-eyed, it means that it has mutations in the same gene. If it's red eyes, it means it has mutations in two different genes. Individually, both genes will give you white eyes, but in this heterozygous state, they will give you a red-eyed blind. So, in this F1 is where you analyze complementation. If the F1 flies are white eyes, we know that white and blanco disrupting the same gene. If, on the other hand, white and blanco mutations disrupt two different genes, they both produce white eyes, then you will get red eyes because you're in effect generating an individual that is heterozygous. You start with this parent that has a homozygous recessive little w mutation, cross to a parent that is homozygous recessive little d mutation, and in the F1 you end up with a heterozygous individual for both the W gene and the B gene, and in effect this individual will be red eyes. So that's how you will determine if they complement or don't complement one another. So the meaning of the word to complement, meaning to help each other. So each one has some defect. If they help each other, they become wild type again. So when a fly gets complemented, if two mutations complement each other and give you wild type flies, it means that the mutations are in two different genes. If two flies do not complement, that means the mutations are in the same gene. So non-complementation indicates mutations in the same gene. Complementation indicates that there are mutations in two different genes. Okay, so if there is a single gene, then you would basically, if the mutations in the same gene, and if you were to map them, those two mutations would basically map in the same spot because they're mutations in the same gene. If, on the other hand, they were mutations in two different genes, they would map somewhere else, and they would be two separate maps, basically. So if the two mutations map to the same site, then they basically become multiple alleles of that same gene, right? Multiple alleles are different forms of the same gene, and that's what you will have generated when you find that they do not complement each other. So, there are two genes, and if there are two genes, then how do we relate the two genes? Well, we relate the two genes very simply. We say there is a precursor, there is an intermediate, and there is a product, and the two genes are working in this pathway of generating this step, right? So if there are two genes, then that's what you would draw out as a form of the pathway. Multiple genes affecting the pathway. Remember the experiments we talked about of Beadle and Tatum. Multiple genes are on the pathway. Each gene generates an enzyme. The enzyme converts from a precursor to an intermediate, to another intermediate, finally to a product. So that's how you would relate the two different genes in a sense. And so if you have two genes, white and blanco, then you would map them. They would be on different chromosomes or on the same chromosomes but very far apart or wherever. But irrespective, there would be two different genes. They would generate two different enzymes.
those enzymes would function in different enzymatic steps in the process. Right? And so they would map differently as well on the chromosome. So, if you have two genes, then the pathway would look like this, or like this. Okay? So, we don't, yes, I know. Here we go. We don't know where the genes would work in this pathway. All you can say is there's a precursor, there is an intermediate, and there is a product. Enzyme 1 would be generated by gene 1, enzyme 2 by gene 2. Alternatively, enzyme 1 could be made by gene 2, and enzyme 2 could be made by gene 1. You cannot order this by the method just by looking at complementation or by doing an epistasis analysis. In this experiment, if you did epistasis, you would get a ratio of 9:7, correct? The ratio we've already talked about earlier. So you would get a 9:7 ratio, but you couldn't place which gene function upstream and which gene function downstream. This is a form of recessive epistasis, but you can't order them because they both, the precursor and the intermediate, have the same phenotype. So at that point, you have to do additional experiments, usually biochemical experiments, to figure things out. Yes?
What is the difference between epistasis test and
Well, in complementation, all you're asking is, do the phenotypes that you have, are they mediated by one gene or two genes? That's the only question you're answering. Epistasis is then trying to order the genes in the pathway, basically. All right? So there you have to take it beyond the F1 generation. You have to go to an F2 generation. In complementation tests, you're analyzing the data at the F1 itself. You don't have to go to an F2 step, basically. So that's the difference. Epistasis, you have to go to a self-cross and then look at the ratios of that self-cross in the F2 project. So, what's the molecular basis of mutations? Right? So let's say you have W1 and W2 and they disrupt the same gene, gene W. What is a gene? Well, it's a piece of DNA, a specific segment of DNA with a very specific sequence that will then code for a protein and an enzyme. And so DNA consists of an array of nucleotides. And so imagine you have the W gene that looks something like this. ATG, there's your start codon. TAA, your stop codon. And then it's got a run of Cs. Okay? So it'll make a protein W. So now, if there's your gene, there's double-stranded DNA, there's your double-stranded DNA, 5' to 3', 5' to 3'. Okay? Maybe in the W1 mutation, you have a mutation right here, such that the C is converted to a T. I'm just giving you a hypothetical situation. So there's one mutation that you have. And so in that individual fly with the W1 mutation, you could imagine a situation where they have a mutation right there. In the W2 mutation, it's the same gene, different individual, same gene, but the mutation is somewhere else on the gene. There it is. It's right here instead of here. So these are two different alleles of the same gene. And they're both going to lead to a mutant protein, and the mutant protein is going to give you white eyes. Right? So what happens during complementation? Why do the two mutations not complement each other? Because here is the cell, here is one copy. This is a diploid organism, so here's one copy. Mutation in this gene, so it doesn't produce a functional protein. And here's the second copy, mutation in this DNA, so it doesn't produce a functional protein. And so you end up with non-complementation. You have two copies, but they don't complement each other because they have mutations in the same gene. They're not mutations at the same site in the same gene, but they're mutations in the same gene such that both of them are inactive. And so they are unable to complement each other. Okay? So now let's make things more complicated. We still have this situation that there are two genes, gene W and gene B. Okay? Let's imagine there's your pathway. But now you can have multiple alleles. You can have two alleles for gene W, and you can have a third mutation that happens in gene B. Gene B also gives you white intermediate. Gene W also gives you a white precursor. And so you have 3 different alleles. And how do you figure out which allele are in which genes? That's the question. How do you figure this out? How do you know that W1 is in gene W, W2 is in gene W, and W3 is in gene B? So what you do is a whole series of different crosses. You start with true breeding flies. That's the first important step. The second point to keep in mind is that you can only do complementation for recessive mutations. Okay? So, what you have to do is basically cross a homozygous recessive W1 fly to a homozygous recessive W2 fly. And you ask, what happens? Does it complement or does it not complement? If W1 and W2 disrupt gene W, the same gene, they will not complement. And so the flies will still be white eyes in the F1 generation. If you cross a W1 fly with a W3 fly, the mutations will complement each other because they're mutations in 2 different genes. And in the F1, you will find red-eyed flies. Okay? So, if you are W1, W1, big B, big B, there's the first fly. W2, W2, big B, big B, there's another fly. And when you make a cross of this, you're going to end up with a situation that looks like this. W1, W2, big B, big B. Right? And this fly will still be white eyes. The reason, because this gene here is still mutated. Both copies are mutated. And so you will stay white eyes. On the other hand, if you have a fly that's W1, W1, big B, as it is, and a big W, but W3, W3 is mutation in this gene, then when you cross these 2 flies, you're going to get an F1 where you have this copy is mutated, but here is a wild type. This copy is mutated, but here is a wild type, and you get a red fly. Okay? So that's complementation. That's the basis of complementation. It's very simple and it's very quick, and the assay immediately gives you an answer in the F1 generation. You do not have to go any further in the experiment. All right, so here are the results that you would get. W1 crossed to W2, non-complementation, because they are mutations in the same gene. And so you represent it as a minus sign. When they don't complement, it's a minus sign. It means mutations are in the same gene. Okay? This is where things get a little confusing. Non-complementation or a minus sign indicates that the mutations are in the same gene. A plus sign indicates complementation, which means that the 2 mutations are in 2 different genes. Okay? And so W1 cross to W3 will give you red eyes flies, and you denote it as a plus. And W2 with W3 will give you red eyes flies, and that will also be denoted as a plus. So, W1 and W2 are 2 different alleles of gene W. They reside in a complementation group. Complementation group is basically another way of saying they are mutations in a particular single gene. Okay? So just a fancy way of calling it a gene. And W3 resides in a completely different gene or a completely different complementation. All right, so supposing you're studying wing shape, and there are 5 mutations for delta wings, D1, D2, D3, D4, and D5. And you want to know how many genes are disrupted by these mutations. Are there 5 different genes that are involved in the formation of the wing? Is there only 1 wing with 5 different alleles? Are there 2 genes, 3 genes? That's the question you need to do and ask. Okay? And so that's what you're going to basically try and figure out. So you basically do a systematic set of crosses. You cross each of these mutant flies with another mutant fly. And systematically work your way through and you build a table. So you first perform a cross of a homozygous D1 fly to a homozygous D2 fly, and you ask the question, in the F1, when you produce a D2 fly, what is the phenotype of this F1? You cannot predict this phenotype. You have to be given the result. or you have to do the experiment and get the result. It's one or the other. Okay? So there's no way of predicting this result. So, is it flat wings or is it delta wings? If they are flat, that means they disrupt two different genes because they are complementing each other. Delta is the mutant phenotype, flat wings is the wild type phenotype. Then you perform the next cross, D1, D1 cross to D3, D3. Again, you ask the question in the F1, when you get a D1, D3 fly, what is the phenotype of this fly, flat or delta? And systematically you build a complementation table. And so you basically have your 5 mutants, D1, D2, D3, D4, D5. And when you cross D1, of course D1 with D1 will give you a negative, right? Because the mutation's in the same gene at the same spot. And then maybe D1 and D2 is also not complementing. D1 and D3 gives you a complementation, D1 and D4 gives you complementation, D1 and D5 does not give you complementation. So, which alleles are in which genes at this point in the experiment? Is D2 an allele in the same gene as D1? Yes. Is D3 an allele in the same gene as D1? No. D4 also isn't, but D5 is. So D1, D2, and D5 are 3 different alleles in the same gene. Now, D3 and D4 are different genes, but we don't know how many different genes. So you already now, by this one cross, indicated that 3 out of the 5 mutations are in one gene. Right? That's the first result you can write. Then you go on to the next cross, D2. You cross D2 to D2, D2 to D3, D2 to D4, and D2 to D5. You don't have to cross D2 to D1 again because you already crossed D1 to D2, which is the same thing. Right? So very often complementation tables are only shown as a triangle. Just the top half, the bottom half is missing because it's just a mirror image of the other. And there you can find, when you cross D2, D2 does not complement D2, D2 does not complement D5, but it complements D3 and D4. When you take D3, D3 is complemented by D1 and D2, but it does not complement D4. So now, D3 and D4 are non-complementing, so they must be 2 different mutations in another gene. So in effect, those 5 mutants that you have are basically in 2 different genes. D1, D2, and D5 are alleles of one gene, and D3 and D4 are alleles of a second gene. And you can, of course, do the experiments with D4 and D5. You build the complete complementation group, and it will be internally consistent. And so from this, you basically will draw out a chart or a result. You'll come to a conclusion, and the conclusion is there is one complementation group for one gene that has the alleles D1, D2, and D5. There is a second complementation group, gene 2, that has the alleles D3 and D4. And so this is how you would determine the number of genes in a pathway. You can imagine a situation where you can have 5 different genes. You may have 10 different mutations in 5 different genes, or 4 different genes, or whatever. But the complementation table will tell you those numbers very quickly. Okay? Now, you cannot figure out the pathway. All you can say is precursor, intermediate product, delta wings, flat wings. That's about it. And you have an enzyme 1 and an enzyme 2, and you have a gene 1 and a gene 2, but you are not sure which way it will work. It can be this in this fashion. Oops. You can have gene 1 and gene 2, or you can have gene 2 and gene 1. You will not be able to answer that question.
Will this using complementation be only for recessive?
Yes, you can only do this for recessives. You cannot do this for dominant mutations. So if you're doing a cross with a dominant mutation, it will always show up as a mutant phenotype, and so you can never physically complement. All right. So this is complementation. It's very simple, very straightforward. You just have to analyze the table and basically start writing a chart and putting them in groups. And the way I always work with them is go line by line. So if I have my complementation here, I'll start with D1. And I'll open a little bracket and I'll say, okay, D1 is in one complementation group, And then, D2 is a minus, so I'll write down D2 as in the same group as this one. And then I'll walk along and say, okay, D5, and I'll write down D5. Then I'll start the next one with D3, and I'll ask where does it complement or not complement, and open a second bracket and write those numbers. So it's very quick, very straightforward. No hidden problems here, basically. The important thing is it's internally consistent. If D1 does not complement D2, D2 will not complement D1. If D1 does not complement D5, then D5 by definition will not complement D2. Because D1 does not complement D2 and D1 does not complement D5, then D5 cannot complement D2 either. All right? Because they're all alleles of the same gene. So they're internally consistent data. In effect, it's like a little puzzle. If I were to give you part of it missing, you should be able to fill it. And there's a homework question that actually makes you go through that process. Any questions on complementation? No. Yes.
Couldn't you have two different mutant alleles that were so close to each other that when they complement, it would appear that they would have a new amino acid produced that would be...
So you're not looking for recombination,
Okay.
right? All you're looking for is in the F1.
Okay, yeah.
You're starting with two breeding flies and you're just bringing the two chromosomes in the F1 together and asking what's the phenotype. It's only in the F2 that you would have those problems if they were very closely linked, etc., etc. Your recombinant progeny would be very rare because they're very close together. But that's in the F2. And that's the beauty of complementation. You don't have to worry about that. You don't have to count 10,000 flies to figure out if the two alleles are very close together or not. You're just looking in the F1 when you're not worried about recombination. Correct? Okay, so now we finally start moving into mutations. We're going to start with chapter 13, which are large mutations, gross chromosomal changes. And then we're going to move into minor mutations, or very tiny mutations, what we call point mutations. And that will be the next lecture. So right now we're going to talk about the gross chromosomal rearrangements. And then we'll go into fine mutations. And once we are in the fine mutations, we'll talk about molecular methods of studying fine mutations. So we'll talk about a lot of different methods and how you analyze tiny mutations in the genome. So we're gradually making our transition now into molecular biology more than genetics or classic genetics. Okay, so let's quick refresher for you guys on mutations. What's a gene? Since we're going to be gradually moving into the area of looking at sequences, I thought I would give you a quick refresher of this. This is a prokaryotic gene, of course, but still the rules are more or less the same in eukaryotes. You have a reading frame for proteins. But that's not sufficient, obviously, for making a protein. You also have to have a sequence of RNA that's upstream and downstream of the coding region of the gene. You guys are familiar with it from your midterm. There's your ribosome binding site. It's present in the RNA, but it's not present in the protein, right? And then you have a transcription start site, and the transcription goes from left to right in this individual case, which means it's reading the bottom strand, the polymerase, as it's moving. If you just have this, it's not going to be made into a protein. You also need the promoter and the enhancer in eukaryotes. And it's only when you have all of these different elements that you're going to be able to make a protein, correct? And so you have to have a daughter box, and you have to have binding sites for various transcription activators. Once you have all of these made, you make an RNA, and if you have a nice ribosome binding site and you have an AUG, you start making a protein, and the protein will be made reading 3 nucleotides at a time of the messenger RNA until you hit a stop point when the ribosome stops making protein. Now, when you are reading the genetic code, here's a double-stranded DNA. Let's imagine 5' to 3', 5' to 3'. You denature the DNA, and RNA is always made 5' to 3'. So you're going to read the bottom strand, and you're going to make RNA in this fashion: AUG, UUU, AAA, UUA, GCCC. Correct? So there's your RNA made. Then that RNA kind of migrates out of the nucleus if you are a eukaryote, into the cytoplasm, binds a ribosome. Obviously, there are additional sequences that I've kind of eliminated here. So here's the RNA, 5' to 3', and then you have the transfer RNAs that migrate in. A transfer RNA that base pairs with AUG, it reads CAU, and it brings along a methionine, followed by AAA, that's phenylalanine, followed by lysine, followed by the stop codon, and you get transcription or translation terminated. And so you get a 3 amino acid peptide. Right there. Okay, the key thing is when you're reading the RNA, you do not leave gaps. Once you start with the methionine, there are no gaps. Correct? Once you initiate at the methionine, you don't skip a nucleotide. You immediately follow 3 by 3 by 3 by 3 until you hit a stop codon. So you don't do something like this. This does not happen. You don't skip this U and take the next 3 nucleotides. Not allowed. The ribosome doesn't allow that to happen. So no gaps and also no overlaps. So you don't have something that looks like this. You don't share this A for the next tRNA. You don't go UAC, AAA, and then ACC. The ribosome does not do that either. So no gaps and no overlaps when you start reading a protein sequence on the ribosome. And there's your genetic code, of course, you guys are familiar with it, and then all the different things that happen there. Okay, so the properties of the code, it's written in a linear form using nucleotides that comprise the messenger RNA, not the anticodons on the tRNA. The code is a triplet. 3 nucleotides specify 1 amino acid, and it's degenerate. There are more than one triplet code specifying a single amino acid. But it is unambiguous most of the time. Each triplet specifies only 1 amino acid. And the code contains stop signs. There are 3 different stop signs, basically. But then the code has no commas, i.e. gaps, and the code is not overlapping. Okay, so with that, now let's talk about mutations. Mutations, basically, most mutations are harmful to a cell and an individual. It's only rare that mutations are beneficial and you basically have them fixed in the population and then they expand if they're beneficial. And they usually expand depending on the phenotype and the evolutionary pressures and natural selection that's going on. A gene with 1 wild type allele is called a monomorphic gene. 1 wild type allele. You can have multiple alleles that are wild type. You guys understand what I'm saying when I say multiple alleles that are wild type? It's because the code is degenerate. So you can have sequences in the gene at the same position or in a different position. You can have 2 different versions of a gene. One version might read AUU in one individual. In the second individual, the gene might go AUC. Correct? It's still wild type because both of these codons will be producing isoleucine or coding for isoleucine. So you'll have 2 different wild type alleles of the gene. Right? They are alleles because they're different forms of the gene, but they are wild type alleles of the gene. And so those are called polymorphic genes. So monomorphic, only 1 wild type allele of the gene. Polymorphic, multiple wild type alleles of the gene. The vast majority of mutant traits are determined by alleles of more than 1 gene in humans. Most of the traits that we study are caused by multiple genes, right? So they are all multifactorial or multigenic. And we briefly talked about it with respect to height and skin color and eye color and things of that nature. Multiple genes affecting a single trait. Okay, a heterogeneous trait is one that may be caused by mutations in more than 1 gene. Right? You don't have to remember all of these terms. I'm just giving you an idea about what's going on with mutations. So human deafness, genetically inherited deafness, is an example of a heterogeneous trait, and it's thought that there are at least 50 genes that play a role in genetic deafness. So a lot of different genes playing a role in Okay, so an important class of mutations are conditional mutations, and we talked about these temperature-sensitive mutations. These are mutations that function normally at under one condition, but under a slightly altered condition, they give you a mutant phenotype. So these are called restrictive conditions and permissive conditions. Under permissive conditions, the gene is functioning normally. In the restrictive conditions, the gene is functioning is not functioning. Okay, and conditional lethal mutations are common.